champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days. And today we are going to finally finish the chapter that is molecular basis of inheritance by the most favorite topic, that is DNA fingerprinting. I know it's very confusing. Even I was confused. I had to go through so many lectures to understand this particular topic when I was studying it. And I have come with a cumulative pattern where I have tried my maximum to make it very, very easy for you. Let's see if you understand or not. Okay, so without further ado, let's just begin. And what did we cover in the previous session, children? We spoke about the Human Genome Project. Now, if you want the PDF for any of the classes that has been already done, you can join our WhatsApp channel. It includes just us, the NEET family, no one else. Now, the number of questions that have come from this chapter has been 6 the previous year, but before that it had been 10, before that it was again 6, and then before that it was 9. So there is an alternating pattern that you see over here. So this year, that is 2025, so that means next year, you can expect more number of questions. Anyways, obviously this is a high weightage chapter. But if you put more of your effort into this chapter, like this chapter and principles of inheritance and variation and things like that, I think there is a high chance that you could score better. Okay, so give your full attention to it, practice lots of questions from this chapter, revise it often because it is a very big chapter. It might confuse your little brain, right? So I'll say have mind maps, do quick revision at times whenever possible. Okay, just swipe through it. Teach someone else. It is a very good procedure to remember a lot of things. Yeah. Now, talking about today's topic, I'm very excited. Let's begin it. So, basically, I was going to call you baby. Listen. So, basically, what is happening? The whole DNA that is there in a cell. Okay, if this is a cell, this is the nucleus, and I say there is DNA strand. There are so many DNA strands. The whole of these things, the bunch, I call it a genome. What do I call it? The whole thing together, I call this as genome. Now, there are different type of DNA sequences in the genome. I say I'm going to categorize into two parts. Okay, I'm going to categorize into two parts. One is bulk DNA and the other one is repetitive DNA, and you don't know nothing about it. You don't know anything about it as of now. You just divide into bulk DNA and repetitive DNA. How did I come to this conclusion that there are two types? Basically, centrifugation was done. Centrifugation, you do understand, when something is being spun in too much speed, okay, at this point of time, according to its density, it is being separated. So it was seen that this particular DNA, this type of DNA, some was getting separated like this and some others were getting separated over here like this. Okay, so one was said to be the bulk DNA and the other was said to be the repetitive DNA. Fine. And now I'm going to tell you one more thing about bulk DNA. Bulk DNA is the one that will be actually coding for proteins. Bulk DNA is actually the one that will be coding for proteins. Whereas when you talk about repetitive DNA, they do not code for anything. They are not going to code for the proteins. So then what's the use? They're just there. They are just there, children. So basically, what do you mean by repetitive DNA? I'll tell you that. Repetitive DNA, as the name suggests, there are repeating units. Okay. So for example, I say there is a base pair that is AATG. Okay, now if I see a strand of DNA, there are so many AATG, then goes on AATG, it just goes on. There are so many DNA sequences that are being repeated again and again and again. And I'll tell you one fun fact about it. So the bulk DNA that we are having, that you and me are having, okay, we almost have 99.9% the same DNA. Okay, 99.9% the same DNA. And it's because of the bulk DNA, because all of us has the same type of bulk DNA. So this 0.1% difference between you and me brings the variation, makes us very different. And who is responsible for that? This guy is responsible for that. So the 0.1 difference that is having, which makes us totally different from each other, is because of this repeated DNA sequence. Now you might wonder how. So basically the thing is, suppose there is one chromosome, my chromosome, okay, this is my chromosome and this is your chromosome, chromosome number 11 of yours, this is your children and this is mine. Fine, I am saying that I have AATG sequences, almost it is repeated 5 times in my DNA. So over here, there are 5 times that this particular thing is repeated in my DNA. But in your case, due to some mutations, due to some changes in your, or due to some changes in mine, anyways, I have just 5, but you have 11 of these things. you will be having 11 of these repeats. So these repeats are going to repeat itself 11 times, but in my case it is just repeating 5 times. It is just repeating 5 times, and I'm telling you, this difference that is happening makes us very unique. This difference that is there in our DNA makes us very, very unique from each other, and this is the reason why we are different as well. Okay, so repetitive DNA is also known as satellite DNA. Okay, so repetitive DNA is also known as satellite DNA, and if you're wondering whether there are different types of satellite DNA, I am pretty sure you're not wondering about that. You might be still confused and know that, but still I'm telling you, there are two different types of satellite DNA. Okay, so we have to see them also, then only you might understand things in a bit more better amount. So over here, satellite DNA I can divide into mini satellite DNA and micro satellite DNA. What do I mean by mini satellite DNA? Over here I am saying the repeats that are there, the length is almost 10 to 100 base pairs. So for example, I can think that I make a AATGCC TTA. How many is there? 1, 2, 3, 4, 5, 6, 7, 8, and then 9 and 10. Okay, this is a sequence. This is a sequence, and this sequence is having how many base pairs, children? Almost 10 of them. Almost 10 of them. And now I am saying that this is getting repeated. This sequence is getting repeated. So here also the same sequence will be there. Here also the same sequence will be there. So this is a repetitive DNA. Now another stance where basically I have AATG. How many sequences are there? 1, 2, 3, 4. I just have 4. Now I am saying that this is getting repeated. This is also getting repeated. How can I differentiate between mini satellite and microsatellite? Both are repeated sequences. Here also this is getting repeated. This is also getting repeated. How can I differentiate between them? Basically here I see that the repeating unit, this particular unit, is having how many base pairs, children? 10 base pairs. This is a long repetitive sequence. But when you compare to this will be what? Short repetitive sequence. Okay, so this is known as microsatellite. Fine, this is known as microsatellite DNA, and this one that you see over here is known as mini satellite DNA. Okay, so mini is not actually mini. Micro, you know, micro is very small. Okay, smaller than mini. So micro is over here, it's very small unit, and then you have mini with almost 10 to 100 base pairs. It can go up to 100 base pairs as well. Now, you have a bit more scientific term for it. I won't say this is exactly known as this, but you have to understand that sometimes people say microsatellites to be short tandem repeats. What do they say? That it is what? Short tandem repeats. Tandem as in it is happening continuously. That is why it is known as tandem. Okay, and short, they are shorter, right? Now over here in this case, they are known as variable number of tandem repeats. Variable number of tandem repeats. So I cannot say that mini satellites are known as this, no. But mostly both of them, it is said that they are associated. Similarly, both of them, it is also said that they are also associated. Okay, so repetitive DNA is fine. Repetitive DNA sequence is fine. And let me tell you, basically all of us might have had 5 repeated sequence. You also have had this thing as 5 in number, but there are certain mutations that happens in us. Okay, some mutations which is not causing a lot of trouble. So mutations can be of 2 types. One that can cause trouble, which would obviously make you have some disease. Okay, or some other type of mutation which is just passed on, which creates variation and which is just passed on from generation to generation. So because of this mutation, the difference has come that I have 5 and you might have 11. Okay, and this is very unique. If I have 5 children and you have 11, this is very unique to you. You will have just 11, I will have just 5. This is unique to me. No one will have that. Okay, now if you suppose that, okay, some other person is also having 5 AATG. 5 AATG. So let me tell you, it's not just AATG that is a repetitive unit. There can be other also. For example, after I stretch this particular thing, the other thing that is getting repeated is GCAA. There is something else that is GCAA, that is the second repeated. Now in this case, what will happen? Basically, probably the person who is having 5, just like me, of AATG sequence, this person will not be having GCAA sequence just like me. For example, if I have GCAA sequence in 2, okay, my GCAA sequence are in 2 amount, but here it will be 5, which again makes me different from this person as well. So everyone has a unique number of repeats and we do not share it with someone else. We all have different number of repeats. Fine. Okay, and this is an example of DNA polymorphism. This is an example of DNA polymorphism. Okay, poly as in many and morphism as in forms. We have so many different forms in ways that we are very different from each other, and when these mutations basically are going to be passed on generation to generation, and we say that almost it is more than 1% of the people who have this mutation, I call this as an example of DNA polymorphism. There is different forms, one have 5, other have 11 and things like that. Okay, cool, works. So this is about the repetitive sequences and things like that. Going ahead, are these sequences of any use? Do you think that repetitive DNA sequence, I told you it's very unique, only some people have the same, I mean like we all have different types of repetitive sequences, the length, the number, everything is kind of different between you and me, that makes us different. But what is the use of it? Is there any use? Basically, I'll tell you, there is use, and that use is if you want to find your parents. If you want to find your parents, at that point of time it becomes useful. Okay, because obviously, for example, your father is passing down 5 repeats, okay, and your mother is passing down 6 repeats. I will be having a total of these many repeats in me. So am I unique with them? I'm not exactly looking like my parents. Why am I not exactly looking like my parents? Because my father is just giving me 5 repeats, but my mother is giving me 6 repeats. Together it's 11 in me. Together I have what? 11. So that makes me different from them. Right? So over here also, mother's DNA might have, for example, one chromosome might have 6, the other chromosome might have 5. Okay, or maybe 6 only. And this chromosome was the one that was passed on to me. So I have 6, but she has what? 12. Okay, if you're getting confused, I can explain to you once more. Listen to me very carefully, okay? So over here, I am saying father is there and mother is there. Okay? Everyone has a pair of chromosome, right? So this is chromosome number 11. This is the pair. This is one pair. This is another pair. I am saying that the father is having 1, 2, 3, 4, 5 repeats in one. Whereas over here, he is having 1, 2, 3, 4, 5, 6 repeats. Fine. In the case of mother, she is having 1, 2, 3 repeats over here in this chromosome. Whereas on this chromosome, she has 1, 2, 3, 4, 5 repeats. Okay. So here if you total it, it is what? 6 plus 5 is 11. Here if you total it, it's 3 plus 1, 2, 3, 4, 5, it is 8. How many repeats are there? 8. Over here, 11. Okay. Now, both of them, when they have to make me, obviously they have to pass half of the chromosome. So suppose they are passing this chromosome. Father is passing this chromosome. Now, how many repeats do I have? 1, 2, 3, 4, 5 from father. And over here from mother, how many do I have? Suppose she is passing me this. So it will be 1, 2, 3, 4, 5. How many of them do I have? 10. I will be having 10. And now if you are supposing this and this will come, children, there will be other variable repeats that will be different from both of them. It won't be exactly the same. There will be other sequences that will be different. I am talking about just one sequence over here right now. So basically at this point of time, okay, with the help of this, I can identify my parents as well. So if my parents have, if the father has 5 repeats like this, I will also have 5 repeats like this. That is, if that comes, that is true. Basically that means that this particular person who is there is my father. Got it? So that is for parental identification. Apart from that, okay, the second scenario is when, okay, is when basically you have some crime going on and there is blood stain, or basically someone's hair follicle is there, and now you have the hair follicle or the person's suspect's blood, suspect, not victims, okay, suspect's blood over there, and you need to find out who did the crime. So you go on matching this with the people who you are doubting. Now you have 3 people in your list and you are doubting 3 of them, and when you match it, you find that this repeat is same like the person who committed the crime, and then you get to know who is the one. We'll get to that as well. Okay, so are these sequences of any use? One is this thing that I was trying to tell you. Okay, so over here I have 10 repeated sequence of GATC on chromosome number 12. That is said by one person. Whereas the other person says I have 2 repeated sequence of GATC on chromosome number 12. So there is difference in them, and this difference makes them different in overall sense, and it will be useful in the case of a process which is known as DNA fingerprinting, about which we are going to study. Just like how our fingerprints are different from each other, similarly in DNA fingerprinting we are focusing on repeated sequence which are also different from each other. We are different from each other on the basis of our repeated sequences. Now, one more thing, if an inheritable mutation is observed in a population at a high frequency more than 0.1%, it is referred to as DNA polymorphism. So sorry, I wrote 1% over here. For DNA polymorphism, it is 0.1. Okay, it is 0.1%. So over here, if the inheritable mutation, something that is going to be inherited, okay, that mutation is observed in a population with a frequency of this, then it is known as DNA polymorphism, and this is an example because lots of people have this. That is what this particular sequence of lots of people are having. This is an example of DNA polymorphism, and it has arisen because of mutation. Cool. Now, starting with DNA printing, so DNA fingerprinting, this is the crime scene. I want to find the person who did the crime. So first thing that I do is, for example, there is blood stain, not of the person who died, from some other blood stain is there. Okay, there is some other blood stain. I took the sample. Okay, I took the sample, and whoever person's sample is that, the DNA is sequenced. Okay, with the help of DNA sequencing mechanism, the DNA has been sequenced, and while sequencing the DNA, it came to the conclusion that the repeated DNA sequence is found to be GGTC. What is the sequence that is being repeated very often and would be very easy for me to recognize? It is GGTC, children. Okay, so please remember this GGTC. And now I have 2 suspects over here, this guy and this girl. These are the suspect. So what did I understand? Basically, in the area where the crime was committed, there was blood stain, okay, or there was hair follicle, whatever you want to take, you can take. Anyways, you are taking a cell and you are going to example. The cell has the DNA, you are going to sequence all the DNA strand over there, and then you get to know that GGTC is the one, that is the sequence that is being repeated. Cool. Now, next thing that I will be doing is, I will be taking their DNA as well. The DNA is isolated. Their DNA will be taken, their DNA is isolated, and after that it is digested with the help of enzymes. As in, basically, if this is a DNA strand, I am cutting it out into smaller fragments with the help of enzymes which are known as restriction endonucleases. What do you call them? Restriction endonucleases. They are going to cleave from inside. Okay. Now, separation of DNA fragments by electrophoresis. After doing that, basically, after cutting the DNA sequences of specific spots, what I am going to do, I am going to run their fragments on electrophoresis, gel electrophoresis. In the case of gel electrophoresis, you know, right, DNA is negatively charged. Okay, so basically it will go to the positive end. It's a gel. Okay, and the gel, you have small wells, and over here the DNA that is negatively charged will be going to the positive end. Okay, and depending on the length of the fragment, it will be divided. So there's a small fragment over here which has 1 repeat. Okay, and this guy is having it. This is guys, and suppose this is the girls. This is 1, 2, 3, 4, 5, 6. We are seeing the number of repeats as well. So the DNA fragments which was small, which has just 1 repeat, is over here. Okay, the DNA fragment which was having 2 repeats is over here. So suppose this guy has had it over here, and then there are 3 repeats over here, then there are 4 over here, then there is 5 over here, and this, talking about the female, she has 2 repeats over here, and then she has 6 repeats over here, and it's, if you see, it's very unique for both of them. It's unique for both of them. Sometimes it can be shared also. This person might also have 3 repeats over here. Do you understand what I am saying? Basically, the DNA is fragmented. Now, so suppose there is GGTC, two of them are there. Okay, this sequence just has 2 repeats. So over there, it will be coming and standing over here. Now, suppose it has 3 repeats, it will be coming and being over here. If we had 4, depending on the length, it will be arranging itself in this particular unit. Now, this is done. This method is known as gel electrophoresis, where according to this, the DNA fragments are being separated. But can you see this DNA fragment, children? Will you be able to visualize this DNA fragment? Chumma, if it is like that, obviously it is not possible to visualize it. So what do? We transfer it into something else so that it becomes visible. Okay. So this next process is known as southern blotting. What do you call it? Southern blotting, or in your textbook it is just written blotting. So let's think of blotting. So what is happening? Basically, we are putting a nitrocellulose sheet or nylon sheet over it and transferring whatever content is there. The DNA, whichever is there, it is being transferred on this particular sheet which I have just put. What is this sheet, children? This is nitrocellulose sheet or nylon sheet. Fine. So the transferring of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. Now that is also done. Okay. After that, what I am going to do? After that, basically comes a twist. So I know that the person's, whoever's sample was over here, it had GGTC. It had GGTC. Now I have to find whether the person over here also, they also have GGTC itself. Okay, they also have GGTC itself. I want to find out that, right? So over here, obviously, if that person also has GGTC, I have to make something which will be complementary to the GGTC, which can go bind there and show the effect. Okay, so we know GGTC is the thing. Okay, now we are going to make a complementary strand. So G, here it will be C. Here also it will be C, here it will be A, here it will be G. So I have made a complementary probe over here. What is this? A probe over here, and this probe, children, is radioactive. This probe that you see over here, it is radioactive. It will show up. Okay, it will show up, it will change, have a different color when you view it under autoradiography machine. Okay, so basically I have made a complementary probe such that this particular probe can go and attach to GGTC sequence if at all it is found over there. Right, now the probe is created, that is the next step. So until now, what did we do? We just transferred everything. The DNA is transferred now onto the nitrocellulose and nylon sheet. It is just kept there. Now I created a probe which is radioactive. Okay, and now I let the probe go and attach to whatever sequence, so complementary sequences is there. Now if there is complementary sequence, it will attach. If there is no complementary sequence, it won't attach, right? Now the scenario is that after I see it under a machine that is known as autoradiography, basically after I do autoradiography, I'll be seeing things like this. Okay, it will be visible like this. So I have the DNA fragments, that is the sample that I had. From this I got this DNA fragments. Okay, this was the DNA fragments. I had the complementary probe, so that is why it is visible. It is radioactive, it is visible. Now over here I see that the GG, you might have got different fragments, but the one that was going and attaching with the probe, or the probe which was basically, wait. So basically what did I do? I had the DNA fragments. I let the probe go and attach to the DNA fragments. So some fragments might not be complementary to it, so obviously it won't attach. Okay, so that is left. I don't want to talk about that. As of now, I am talking about the fragments which have the code, which has the repetitive sequence, that is GGTC. Okay, I gave the probe, that is what? CCAG. This was the probe that I was having. So this probe went and attached, and now all those things are being visible which have the attached probe. Okay, so suppose this also has attached probe, this also has attached probe, this also has complementary to the probe. All of them, they are complementary to the probe. And now when I find that, okay, there is one segment like this, there is another fragment like this, there is another fragment like this. Over here I see these are the fragments that I got. Okay, and CCAG is complementary to all of them, which is why it is visible under autoradiography. And now I see this was a sample that I had. You tell me, is it matching to this or is it matching to this? This is 1 and this is 2. Onto which is it matching more? Over here, if I see this strand is there, this strand is also there, this strand is there, this strand is also there, this is there, this is also there. It's the exact same thing that I got over here. So you tell me, the person who committed the crime, who is he or she? It is a she, and she did the crime, children. Okay, I'll just repeat it once more. Listen to me very carefully. I got the sample, I sequenced it, okay, and I found that GGTC was the thing that was getting repeated. Now I had to find out the exact person who have exact number of repeats just like this particular sample. So what did I do? I took both of their DNA, I isolated it, I cut it with the help of restriction endonucleases, which just cut the sequences which has this. which has this particular sequence, then only it's very programmed method. Okay, I cut those sequences. Now the next thing is that I did separation of the fragments by electrophoresis. So the one that had smaller came over here, one that had bigger were over there. They arranged themselves. Now I cannot see it. So what do? Basically, I took a blotting paper that was made up of nitrocellulose or nylon, and then I transferred everything over there. Still I'm not able to see anything. So what do? I made a probe. Okay, I made a probe over here, and this is the probe that I am having, and this is a radioactive probe. It will show the color of when under the autoradiography machine it's there. Okay, so what will happen? I made a probe which is complementary to this particular thing. Now whenever I see this particular thing, this probe will go and attach over there, right? This probe will go and attach over there, and it will blink off. So I will be able to see that, okay, this is the area which is having exact complementary base pair, right? Now when I did this, I found that the sample that I had, okay, it also got attached to CCAG, and which is why it was visible. All of them are visible because they are having attachment to this. Now I got to know that basically this guy has this profile, whereas this particular female that is there had this particular profile. These many sequences were there which were repeated, and this is exactly similar to what I found in the sample. So who is the culprit, children? She is the culprit, and this process is known as DNA fingerprinting, and you will be able to see this only using autoradiography. Otherwise you would not be able to visualize this particular thing. Okay, now I think you might have got certain idea. Now this is the thing that is given in your book. In your book, what is given? So pattern from the father's chromosome, from the mother's chromosome, you have got this thing. So you saw over here that on chromosome number 7 you have 3 from the father and 4 from the mother. Similarly on this you have so many from here and so many from here. This is from individual number B, and this is from individual number A. Same chromosome you are taking. Okay, you have taken so many chromosomes, and all the chromosomes have different number of repeats. Obviously you are getting from father and mother both, so obviously in your case also you will be different from your parents. Okay, so you have got that, and now you have made it run. You have made it run, and then you found out that basically the sequences were arranging in this pattern. The sequences were arranging in this pattern, and when you took the sample of the person who did the crime, it came out their sequencing is like this. Their sequencing is like this. Over here they have colored, so making things more understandable. Is the same thing. You're talking about one repetitive sequence only. There's only one repetitive sequence you're talking about. Suppose it is AATG. Okay, depending on the length they have differentiated. Okay, now over here you saw that basically what is happening, children, this and this, if you see, they are exactly similar. This and this, if you see, they are exactly similar, because the parents obviously gave this, so I have taken both the parents' chromosome. Okay, both the parents' chromosomes things are there in me, so obviously I have taken both of theirs and put it over here. This particular sample will also have both my parents, because my cell is being taken, so my cell has both the parents' DNA. So if I see this and this is exactly the same, my dear children, it will be the exactly same. Got it? And this is how I can find that who did the particular crime. Works? Is it okay? So over here, to make it less difficult for you to understand, I can say, where did that go? Okay, I think I removed it. Basically, when I was talking about the father and the mother's DNA for finding out who is the parent and everything like that, you need to number the counts. You do not need to total it out. For example, the father has 8. Okay, father has 8, and this chromosome of father has 7. Just count 8 and 7 separate. Do not need to calculate it together and then need to find it. Okay, so this 8 one will be going over here, 7 one will be going over here in the electrophoresis. Now if you have 2 of them, basically we get to know this is father's. Okay, this is the father's thing. Fine, got it? Now I think that is up. That's all for DNA fingerprinting. If you have still not understood, you can tell me, okay, because I know it's kind of confusing topic. Going ahead, we have to solve the questions, children. So that is the first question that I have for you. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? Restriction enzymes basically were used to cut, right? DNA hybridization, I see the complementary probe was there for obviously coming together with the other DNA strand, right? So it is also there. Polymerase chain reaction, oh, I didn't mention about that. Okay, one more thing. Basically, digestion is done with the help of restriction endonucleases. After that, the number of this DNA is also being increased. Okay, or I think before that, over here, the DNA is isolated at this point of time. PSCR is done, that is polymerase chain reaction, such that the DNA, the amount of DNA could be increased, and we could make it work with the restriction endonucleases. So over here I am increasing the amount of DNA, and then restriction endonucleases cut them, and we have multiple fragments over there. So obviously I cannot work with one DNA sequence, I would need multiple sequences, which is why I am doing this. Okay, so PCR is also there. Okay, so you have restriction enzymes, you have DNA hybridization, you have polymerase chain reaction, and then you have zinc finger analysis. Do you need it here? Do you think that you need it over here? You tell me. You tell me the answer, okay? I'm not telling the answer, you have to tell me the answer. So restriction enzymes, I think is there, right? DNA hybridization, again I think is there. Polymerase reaction, again I think is there. Zinc finger analysis, is it there or not? Or do you think the question itself is wrong? You can have that as well. There is challenging questions as well, that the question itself comes to be wrong. So you can tell me that. Now, second question, one of the most frequently used techniques in DNA fingerprinting is VNTR, SSCP, SCAR or AFLP. You have to tell me again. So if you have listened to my lecture very carefully, you might know that there is one term among them which we have discussed in this particular class. So think about that. Okay, I will solve it later on. Now third question, what is that forms the basis of DNA fingerprinting? The relative proportions of purines and pyrimidines in DNA, the relative difference in the DNA occurrence in blood, skin and saliva. Do you think that the difference is there in the DNA of our blood, skin and saliva? No. All of our cells has the same set of DNA. None of the cells have different set of DNA. All of them have the same set. Whether it be blood, whether it be skin, whether it be saliva, it's of our body. If it's of our body, it has to be having the same DNA content. Okay. The next thing, the relative amount of DNA in the ridges and the grooves of the fingerprints. Do you think, is it because DNA fingerprinting means like the ridges and the grooves that you see over here, okay, the DNA which is there over here, which makes the whole difference, and this is how the DNA fingerprinting works? Do you think like that? Next thing, satellite DNA occurring as highly repeated short DNA segments. What is the correct answer for this? Again, very direct question, very easy question, but people who do not know DNA fingerprinting at all might face difficulty in knowing the answer as well. Now, fourth question. DNA fingerprinting refers to molecular analysis of profiles of DNA samples, analysis of DNA samples using imprinting devices, technique used for molecular analysis of different specimens of DNA, technique used for identification of fingerprints of individuals. Tell me. DNA fingerprinting refers to what? What is the correct answer? You can put it down in the comment section. Yeah, going ahead. Fifth question, DNA fingerprinting is based on the principle of identifying dash sequences of DNA. This I can very easily say, but I want you to tell me. DNA fingerprinting is based on the principle of identifying dash sequences of DNA. Satellite, variable, mutated, repetitive. What is the most apt answer for this? What is the most apt answer for this? This will be repetitive, even though you might feel that this and this everything is connected in some other way. But yes, we are talking about repetitive DNA sequences. You call them satellite DNA. That is different. But yes, it is repetitive DNA sequences. Okay, cool. Or if you want to be more specific, this is not a NEET question. This was just put down there. So I just put it down here as well. Now, let's solve the questions which were asked. So, which of the following is not required for any of the techniques of DNA fingerprinting available at present? It is zinc finger analysis, which is not required. Rest all of them you saw, it is involved in the process of DNA fingerprinting. Now, one of the most frequently used techniques in DNA fingerprinting is VNTR, children. What is VNTR? I told you, it finds the variable number of tandem repeats. Variable number of tandem repeats, that is mini satellites repeats. Cool. Now, what is that forms the basis of DNA fingerprinting? The relative proportions of purines and pyrimidines? No, children. The relative difference in DNA occurrence in blood, skin and saliva? No. Everything is same. The relative amount of DNA in the ridges and the grooves of fingerprints? No. Satellite DNA occurring as highly repeated short DNA segments. This is the correct answer. Cool. Works? Now next question. DNA fingerprinting refers to what? Molecular analysis of profiles of DNA sample, analysis of DNA samples using imprinting devices. Okay. Techniques used for molecular analysis of different specimens of DNA, technique used for identification of fingerprints of individual. Obviously this is not the answer. Obviously this is not the answer. Children, just a second, okay? Over here, just give me a second. Yeah, so D is not the answer. Now, technique used for molecular analysis of different specimens of DNA. Do you mean your DNA fingerprinting means that? No, children. There are no techniques used for, you cannot combine DNA fingerprinting as techniques or something like that, okay? Now, analysis of DNA sample using imprinting devices, no. Molecular analysis of profiles of DNA samples, this is the thing. So, you are analyzing the DNA strand from the molecular basis. You are looking for each nucleotide, the repetitive sequence, and then you are analyzing things, okay? So, this is the correct answer for the same. Now, going ahead, so we have completed DNA fingerprinting, and as of now, if you still have doubt, I understand, you can still ask me. I can make something out of it. I'll try to teach you as much as possible, okay, for this particular topic. And once you're done with that, okay, once you're done with that, let's begin the next chapter. So are you ready for evolution? So evolution will be the next chapter. So according to weightage, we are going. So in 12th, after this, we will be starting with biotechnology and principles and procedures. But as of now, we will be going with evolution. So are you ready for that? I hope you are ready for that. And if you think that this particular video was useful for you, then do not forget to subscribe to our channel. And if you still have doubts, as I said, please feel very free to ask me the doubt in the comment section. I will help you out for sure. And I wish you have a very, very good day. Bye-bye. Take care.